invited me here. It's a pleasure to tell you some stories that I have been thinking about recently. To begin with, my two co-authors on this work are here, Jean-François Joanny from France, from Paris in France, and my student at NYU, Kate Sanford, who just recently defended his PhD on this subject. So what I want to tell you about, I will have to find out how to turn the slides, because I'm used to do it distantly, but my computer for some reason didn't like to work here. So the big problem that I'm thinking about is chromosomes. You know, chromosomes in our body, in every cell of our body, though there are particles which house DNA, and all genes are there, and everything is there, so it's very important. And for some reason, biologists know for many years already, many decades, for about a century, that chromatin, this material of chromosomes, is somehow segregated into so-called heterochromatin and euchromatin. And the different parts of DNA in different cells go to either euchromatin or heterochromatin, depending on which cell it is, whether it is brain cell or liver cell or some other cell. And the fact of the matter is, heterochromatin is the part of the DNA which in the given cell is used. It contains all the genes which are expressed in the given cell and genes which are silent, and you know that DNA is the same in all cells, and different cells need different functions, so they need different proteins. And so euchromatin is the passive stuff which is not used in the given cell. But they are segregated somehow in space. So why could it be? And I have a hypothesis about it, which may or may not be right, but whether it's right or wrong remains to be seen. But I think on the way we discovered quite a few interesting physics stories, so I am about to tell you these physics stories, which are motivated by this picture of segregation of chromatin. I will come back at the end to chromatin, but for now, I forgot again that I have to do it like this. Okay, so about phase segregation. I want to tell you that phase segregation, there are two types of phase segregation, as shown in these two pictures. In one case, you basically have the same molecules in both phases. In this case, phases are liquid and solid, but it could be liquid and vapor and gas and so on. But it's the same molecules. Chemical composition of both phases is the same. In this case, it's very different. It's segregation of oil and water, and this is a different type of thing. So there are two different things, two different phase segregations, and I will call them A from A and A from B. So in my subsequent slides, please pay attention to this red signs with yellow letters, A from A or A from B. Every time there will be A from A or A from B. So how do we describe this type of thing in statistical mechanics? And what may drive this phase segregations, in this case between A and B? So one possibility is that different molecules interact in different ways. For instance, water, so quote unquote, likes water, and oil likes oil, but they hate each other because they have different dipole moments. So in that case, what happens is you take energy of AB minus energy of A with A minus energy with B with B, and this is the parameter which you have to compare to see if the thing segregates or not. In this case, it's not interaction. There are no interactions, basically. Those are some nuts or something like that. So in that case, segregation between big particles and small particles is entirely based on volume. So in this case, segregation is driven by energy part of free energy, and in this case, it's driven by entropy part of free energy. In general, you may, if this is pairwise dominated, you may write similar formula for virial coefficients instead of energies and volumes. But that's the story of A separating from B.
Why do the large particles float in one case and sink in the other case? Should they occur the same way?
In this example, this is also the question of gravity, who of them is heavier. So it's the same story, why oil goes above water and not other way around. There is a separate story, and that part of my talk is a little bit of a side branch about chirality. In that case, we are talking about phase segregation based on shape, I would say, not size, not interaction, because of course in enantiomers, these type of molecules, they interact the same way, they have the same size, same volume, same everything, except different incompatible shapes. And they form mirror symmetric crystals, which means they phase segregate based on their shapes. And this is peculiar. And in general, this is a very, very interesting subject of a different talk, but I cannot resist, since I mentioned it, I cannot resist showing to you two slides where something similar to phase segregation happens in completely non-physical setting. So when you go home, you will drive on the right side of the road. But in many countries, people drive on the left side of the road. So I would say that this segregation of the map into left driving and right driving countries is similar to segregation of the Ising model into domains of spin up and domains of spin down. And there is coarsening as time progresses. There is coarsening in Ising model. There is similar coarsening. Here, for instance, Sweden recently switched from, half a century ago, switched from left driving to right driving. And presently, you can see that most of left driving countries are on islands. Well, India is obvious counterexample, but India is not really counterexample because there are not many roads on which you can drive from India to outside countries. And therefore, this domain is uncoupled to other parts of the system. From that point of view, it is interesting to compare this map to another map where I show to you left and right-sided railroads. And railroads, this Ising model relaxes much slower than the regular roads. And you see that, for instance, Western Europe, not only Sweden, but Western Europe and even Portugal remain left-sided in terms of railroads, although they switched to right side in terms of regular roads. So it's just for fun. This is phase segregation based on chirality. But I want now to switch gears and to go to real subject of my talk, which is phase segregation based on activity. So it is the subject of non-equilibrium statistical physics, which is very important for biological applications. And in that case, there are many examples where active systems, such as actively moving animals, fishes, birds, and so on, they form something that looks more or less like a drop of condensed phase surrounded by much more dilute region, like here or like there. This is the case of A segregating from A. You have more condensed phase of the same particles and less condensed phase of the same particles. And there are many works on that type. And this picture I brought up because when I was looking on the web for pictures of this sort, I came across this one, and this is very cute, although it has nothing to do with the subject of my talk. Okay, but I mostly wanted to talk to you not about macroscopic creatures such as fishes or birds, but about microscopic, since I'm thinking about chromatin eventually, so I want to think about active particles of microscopic nature. For instance, one such microscopic active particle, which is fundamentally out of thermodynamic equilibrium, would be bacteria. Bacteria do something, if you look in the microscope, what they do is they go trajectories like this. Okay? So that looks, to anybody, it looks like a random walk. So it looks like they move the same, pretty much the same way as, let's say, colloidal particles of few micron size, which are subject to Brownian motion, so they undergo Brownian motion. So who cares if they eat their sugars or they don't eat their sugars? They just perform random walk, so they should behave like dead particles performing random walk, like any colloidal particles. In reality, they do something very different. They do not behave like random walk. They look like random walkers, but they do not behave like passive random walking particles. They do something very different. Here is one example, experimental example of what they do. Imagine a box with divider like this. So this is increased picture of this divider. So the gaps between the doors between these two compartments are arranged like funnels. So it looks like it is, naively, it looks like it's easier to go this way than to go that way. Because if you go this way from left to right, you are sort of funneled into the door. If you go from right to left, you probably have a chance to get stuck in this corner. But you know that in statistical mechanics, the distribution of particles in that part and in that part has nothing to do with the shape of this divider. You just write partition sum, which has nothing to do with your divider and its shape, and it will tell you that, of course, density of particles here and here will be identical. And indeed, if you take dead particles, then the number of particles on both sides is the same. But if you take live bacteria, then no, live bacteria prefer to be on this side. Moreover, later experiments showed that there are two types of bacteria, some reason called pushers and pullers, two types of bacteria, and some of them will accumulate on this side, and another will accumulate on that side. Okay, so they definitely violate detailed bounds. They do something very unusual. Even more striking, you can do with bacteria this trick. Imagine that you take a very tiny little wheel like that and place it in the liquid, or in the liquid with some colloidal particles. Would colloidal particle rotate this turbine due to their thermal motion? What do you think? Colloidal particles. Let's ask students. What students think? With colloidal particles, will turn this turbine in some direction or not? Let's do vote. We are in a democratic country, so we should solve this by voting. Raise your hand if you think this wheel will rotate clockwise. Okay, One person says, think so. Two persons, three persons. Okay? If this will rotate counterclockwise. One person, two persons, three, four. Okay. Who thinks it will not rotate at all? Okay. It's interesting. Should we allow professors to what? Okay, so the answer with colloidal particles, with equilibrium system, it will not rotate at all. If it could rotate with equilibrium system, then you could, of course, attach electric generator to it and receive electricity from nothing, from heat energy, which, as you know, is not possible because of second law of thermodynamics. So with that colloidal particles, this will not rotate at all. No way. However, with bacteria, it does rotate. And as you can guess from the previous example, depending on the type of bacteria, it can rotate either clockwise or counterclockwise, depending on what kind of bacteria you have. Okay, so that means you can construct such machine, but it will work not on the expense of thermal energy of your solution, but on the expense of chemical energy of some sugars which are consumed by bacteria. So that means active particles are really, really, really something unusual. Now there is something, this is actually a movie, which is artificial model. This is artificial model of particles due to these authors. And these particles, what they do is if there is a light, then they catalyze certain chemical reactions. And because of that chemical reaction, they move in certain direction. Okay, so they are like bacteria, but sort of artificial bacteria. And they also violate detailed balance, and you will see what they do. They do something funny. The particles themselves are inert, but when you shine light, they catalyze chemical reaction, and therefore they become active. And so light is on, and you see they swim and they form phase segregation, A from A. There are some particles which remain in sort of gas phase, but other particles assemble. Now light is off, and they diffuse away, they dissolve back. And therefore, this aggregation, light is on, and again they go and aggregate, they form phase segregate in this A from A style.
And when you turn on light, the motion, the direction of motion is still random?
Yes, the direction of motion is random. The only difference when there is light, then they continue moving, they are more persistent. They less frequently change direction. When there is no light, they more frequently change direction. They essentially change direction all the time, like regular Brownian particles. When there is light, they are stubborn. They become stubborn. They persistently move in one state, then they turn, but they are more stubborn. And that is sufficient for them to do this trick of phase separation. Okay, so let us try. By the way, I am theoretical physicist, so I like to consume life through equations. So I will show to you a few equations, but don't be scared. I will not force you to. And by the way, there will be no exam at the end. So normally, Brownian motion is described by this equation, which is called Langevin equation. And this is very simple thing. It tells you that friction force, which is if friction coefficient multiplied by velocity, is friction force, equals the force which pulls your particle, which means, for instance, gravity, plus the force of random kicks. This is random kicks. And this is Langevin equation, and this force has something to do with temperature. So the fact that this force has to do with temperature is related to the fact that it contains diffusion coefficient. And in equilibrium, Mr. Einstein told us that diffusion coefficient is related to friction coefficient through this simple formula. This is Einstein formula. Okay, so this is temperature and friction. The bigger is friction, the smaller is diffusion coefficient. The bigger is temperature, the more actively they diffuse. Very natural thing. And by the way, keep in mind that this paper where Einstein showed that is the most cited paper by Einstein. By far more cited than all this relativity theory, general relativity, and all this other, I wanted to say crap, but I should say all other wonderful things. So this is the most cited. So in equilibrium, this is true. But I told you what I want to study is I want to study phase separation away from equilibrium. So I should violate some of the prescriptions of regular equilibrium statistical mechanics. And what I want to violate is I want to violate this. I want to consider particles which have friction according to their sizes, like bacteria. They have friction according to their sizes, because the bigger, the more friction. But they have diffusion coefficient due to something else, due to the fact that they are active, they are swimming, they have motors, they eat sugars, they do all sorts of wonderful things. And because of that, their diffusion coefficient is bigger than what Einstein prescribed. Let us see. Sorry?
What's the meaning of the delta function?
Oh, that's excellent question. Thank you. You know what for experienced speaker, when the experienced speaker tells you that this is a very good question, you know what that means? That means he knows the answer. So the meaning of delta function is that kicks, random kicks, at even very close time moments are independent from each other. Kicks are provided by other molecules. The number of molecules around is so large that these kicks from this molecule, from that molecule, they are independent. In fact, of course, this delta function is every delta function in physics is somewhat smeared, and this one is smeared by the time of molecular collision, which is very, very small. It's like femtosecond. But this is indeed very important. And by the way, there is another interesting direction when we consider random forces which are correlated over some finite amount of time, which may be true in some biological systems. And that also leads to interesting consequences. So this is the story. Okay, so we can simulate this. We can simulate that and look at the title of this paper, binary mixture of particles with different diffusivities. Different diffusivities exactly means that orange particles and blue particles violate this Einstein relation in different ways. Okay, so they have the same frictions, they have the same size, the same friction, but they have different diffusivities. And you see that with time, this looks very much like the distribution of domains in the Ising model, or if you want, the distribution of left and right driving countries. Okay, so indeed they do segregate. Particles with large diffusivity segregate from particles with small diffusivity. Despite the fact that they interact the same way, they have the same size, the only difference is that one of them moves more actively than the other. How can it possibly be that active motion or level of activity of motion leads them to segregate? Here is another simulation by another group which approached it differently. Instead of looking at many particles, they looked at just two. There are two in this large bus of passive particles, which are blue in this case. You have two active particles, which they call it hot, and they attract. They prefer to come closer, and there is some correlation function and so on. You can make it quantitative, so they basically attract. So now the question is reduced to the following. Suppose you have two particles. Suppose you have two particles, and they are different in terms of their activity. Now, my A and B are not different sizes, not different interactions, nothing like that, different activities. One of them is active, more active, and another is more passive. So how do they live? What happens to them? Okay, so let's take it seriously, and let's consider this situation. So what you have is still the same Lunge equation. You have friction force, friction coefficient multiplied by velocity is the force which acts on particle A. And suppose they have some potential energy U, they interact somehow maybe by potential U, and then there is this the force which acts on particle A. But then there are kicks, random kicks, with temperature TA. Let's say A are hot particles, so TA is larger of the two temperatures. And there is identically looking equation for B, same thing, except different temperature. Okay, so what would that give us? What would these two equations give us? Okay, so I reproduce here these two equations, and this is the heaviest mathematics slide. But let us exercise a little bit. Let's take U if they interact with some potential.
External potential?
It may be external potential. It may be the potential of interaction between these two particles. I have two particles, and they may interact. There may be some spring between them, or some van der Waals interaction, or anything.
But that interaction doesn't depend on their relative positions? I'm just trying to understand this.
Oh, yes, interaction may depend on their relative position. Relative position will be R, xi, xa minus xb. So this will be the derivative with respect to xa. This will be derivative with respect to xb. They will be the same except the sign, which is the third Newton's law. In that case, if U is not external potential but interaction, these two forces are equal and opposite in sign. But let's just exercise in algebra. Let's subtract one from the other. Subtract one equation from the other. So this will give you same zeta, difference in velocities, that would be relative velocity, right? This will give you twice the force, because these two forces are, according to third Newton's law, they are opposite in sign, twice the force. And this thing, what you do is you add two random noises. So you obtain a new random noise whose amplitude is the square root of the sum of squares. It's like adding random measurement errors. So what you obtain is you obtain a regular Langevin equation, except potential now has factor 2, and temperature is the sum of temperatures. So it will come to, with time, it will come to Boltzmann distribution, except what it is. This is very strange temperature. So it's completely non-equilibrium system, totally non-equilibrium system, but it comes to something like a Boltzmann equilibrium with average temperature. This is very, very, very strange. I call it Boltzmann distribution. I call it actually Boltzmann distribution in quotation marks, because Boltzmann, of course, if he were to live today and to see this distribution, he would have killed me and said, no, 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 that's not me. I'm not responsible for this stupidity. No way. So this is something like a Boltzmann distribution, but this is something very unusual. And so I skip this. And what is the consequences of this Boltzmann distribution, quasi-Boltzmann distribution? The consequences are best seen on this graph. What is this graph? Remember, I'm talking about two particles in one-dimensional system. So I have coordinate of one particle, coordinate of the other particle, and then every state of my system is some point on this plane. So I discovered that if I look at the currents of probability here, then I discovered that these currents of probability form loops like that. Which means there is no detailed balance. The system goes in one direction differently than to the other direction. It is similar to these bacteria which go through the funnel-shaped gates in one direction easier than in the other direction. So you can imagine that there are some funnel-shaped gates which force my particles to go like that. You can understand this by thinking about classical Greek Zeno's paradox when Achille, or whoever was it, I forgot, was changing a turtle. And in this case, the idea is very simple. You have a turtle, which is cold particle, passive particle, and you have some body who is active particle, which is driven by higher temperature. So the higher temperature guy runs fast. Of course, it runs randomly because it is driven by random kicks. It runs randomly back and right and left, but since he rides very fast, very frequently he would hit the slow guy. So for instance, if the slow guy is over there and I am running back and forth, I will eventually chase him and push him. But he, over there, he has no chance to push me because he is slow. There is no chance that this turtle will ever hit this running guy. So that means when you're in this diagram, when XA is bigger than XB, that means B particle pushes A particle and they go together in this direction. Then they somehow flip and then they go back together and so on. So basically, this whole story is about how fast particle pushes the slow one, and that violates detailed balance. And as you will see in a second, or a couple seconds, it leads to segregation. Maybe I will show to you before segregation. I want to show to you that this all has very, very practical applications, these considerations of detailed balance, have very practical applications. So this is just a better drawing of the same model where you have two particles and you have loops of probability current, but then you can also look at very practical questions. So in this paper by Fred McIntosh and his co-workers, I have nothing to do with this paper. I'm showing to you paper of other people. I am not author, nobody there. So just no, I'm not claiming anything. I'm not even claiming that they are right, although I think they are. But this is the following story. They look at certain motion of some biological object, some flagella. Doesn't matter what that is. But they want to know whether this object is actively moving or passively moving. So it flaps like that. Or you can imagine that there is, I don't know, a piglet who does something like that with its tail. Is it active or it's just Brownian motion? How do you know? So they suggest the following answer. It's a beautiful, interesting answer. So they expand this shape of this object in 3 Fourier modes. They do Fourier decomposition and look at 3 modes. One mode is like that, another mode like that, and another mode like that. 3 modes. Yeah, it takes 3 modes. And those are 3 coordinates. And then they take movie of this motion and look how this movie is projected in terms of these 3 coordinates. And they see that there is a current of probability. That current of probability means that there is no detailed balance, and that means it is an active motion. No chance that it is a Brownian motion. It is an active motion. It's very, very sensitive test. So if you see something that you don't know whether it's active or passive, this is the test choice for you. Be careful though, because in some projections, it may look almost like there is detailed balance because of projection. But in the other projection, it's very much a loopy current. A loopy current means no detailed balance, no, okay. So I told you the other slide was the most mathematical, therefore I am forced to skip this one. But I will show to you this one. So in the end of the day, if you go through mathematics, based on this, I told you Boltzmann distribution for two particles, if you go through this mathematics and you turn the handle of the meat grinder for a while, then you discover this phase diagram. What this phase diagram means is the following. Every point here, it is a 3-component system. You have some fraction of A particles, some fraction of B particles, and some fraction of solvent. Together, they give you 1. It's volume fractions of different particles. And in this triangle, every point represents certain composition of these 3 concentrations. Phi B is the distance to this side, phi A is the distance to that side, and phi solvent is accordingly the distance to the base. Then there is this gray area in phase diagram where the system is completely unstable. Then there is spinodal, and then there is also the line of constant pressure, which means the line at which 2 different phases will coexist. So this phase will coexist with that phase. Okay, so because of this strange Boltzmann distribution, we discover that there is indeed the possibility of phase segregation. When the system will tend to phase segregate, if these 2 temperatures, the whole story is driven by the difference in activities. And activities here are proxied by different temperatures. They're not real temperatures because the system is out of equilibrium, but those are proxies for activities. So how much activities of the particles should be different for the system to phase separate? They should be quite significantly different. You see there are strange numbers here, but basically inside this interval of ratios of temperatures between 300 and 34, the system is stable. So in principle, you can drive phase segregation like that, but it's difficult. You have to really, really, really drive your systems through. So factor of 13 temperatures is too much.
If you put equal temperatures, that satisfies the inequality? Is there a mistake in the slide?
No, because it says that the system is, the system, it should say the system is stable if inside this interval the system is stable. Yeah, I'm sorry. If you have a marker, we can change it on the screen. Oh, maybe it's because of the different computer. Yes, thank you. It's a mistake. Okay, so this is the same diagram, bigger. And this slide also shows something interesting. So you can drive segregation by contrast of volumes. That we started from nuts. This is nothing new. You can drive segregation by contrast of temperatures, by the difference of temperatures. But you can also drive it by contrast of frictions. This is quite interesting because in equilibrium statistical mechanics, friction is absent. There is no such thing as friction because how equilibrium statistical mechanics works. You write Z equals the sum or integral of Boltzmann exponentials. That's it. No friction, no place for friction. Whatever the friction, it has nothing to do with equilibrium statistics. But it's not equilibrium. So if frictions are sufficiently different, then you can, if you are above this surface, you can drive segregation, which is just an interesting note. Okay, so far, let's summarize. So far, we discovered that indeed, if particles are different in terms of their activity, they may segregate, but it requires much effort. One of them has to be much, 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 much more active than the other to drive segregation. So how can we go to something more down to earth? And for that, a good prescription is to use, instead of separate particles, take particles which are connected in two chains. Instead of particles, take chains. Take chains of particles, chains of monomers, chains of particles. Polymerized matter, I call it. So if you go back to equilibrium statistical mechanics and consider polymerized matter, which is polymer physics, essentially. Yes, there is phase segregation there too, of course. And phase segregation there has very interesting properties because you can have many different interesting phases. I will not talk about that, but it's extremely interesting subject in its own right. When you segregate red from blue, but red and blue are somehow connected by polymer chains, so they cannot segregate far, and they instead segregate into microphases, microphase segregation. But I want to concentrate on a different aspect. We discussed that this is the property when segregation is driven by difference in interaction energies. This combination of energies is deciding whether the system wants to segregate or not. So I would argue that when matter is polymerized, then critical value of this characteristic parameter gets reduced by a factor of 1 over n, n being the number of monomers or units in the polymer chain. Why it is so? To understand it, you don't have to know much. It's very easy to understand. Just think about a regular phase segregation between oil and water. What happens in that case? When you separate oil from water, what you gain is you gain unfavorable energy of interaction between oil and water. They don't like it. So you gain it, okay? But then what do you lose? You lose entropy in the amount of kBT log 2, roughly speaking, log 2. If the volumes of two phases are the same, it's log 2. Otherwise, it's log of ratio of volumes or something like that. So when energy loss is compensated by entropy gain, you phase segregate. That's what it is, the competition of energy and entropy. When you have polymers, it's the same story, except energy that you lose or that you gain, depending on the direction, energy difference between two phases is proportional to the number of monomers. While when you move one chain here and one chain there, entropy is driven by the number of chains. Right? The fact, for instance, that in this half of the room, almost all of the students see it in the back half, the entropy that you lose by doing that is proportional to the number of students. It's not proportional to the number of molecules of which the students are composed. It's only the number of students, right? So similarly, number of chains. Number of chains is n times smaller than the number of molecules, or monomers in that case, which is why critical value of this chi is proportional to 1 over n. In reality, it's very difficult to mix different polymers. They almost never mix. It's a big industrial problem to mix polymers, but they almost never mix because of that. So therefore, let's try to think what happens if we take polymers with different activities. Let's take polymer chain and imagine that 1/2 of this polymer chain consists of actively moving particles with high, which have access to thermostat with high temperature, and another half of polymer chain consists of other chains which don't have access to high temperature. And then they do segregate, as this picture shows, and this is a PRL paper by Jan Schmrek and Kurt Kremer, and you see it does segregate, and it does not require temperature contrast to be 35 or anything. It's temperature contrast of the level of a few percent, and you already have this type of segregation. How am I doing in terms of time? I think I should wrap up, and I'm approaching the end of my story. Indeed. Let me skip that, and let me come back to hetero and euchromatin. So basically, my hypothesis, I think you can already guess what is my hypothesis. My hypothesis is that heterochromatin is subject to all sorts of intracellular processes. There is RNA polymerization, there is this and that, and so on. There is lots of activity happening around heterochromatin. It is a little not entirely clear that this can be described by higher temperature, but to some qualitative degree, maybe it's similar. So maybe they are segregated. Heterochromatin is segregated from euchromatin because heterochromatin is more active. It is polymerized matter. DNA is a polymer, you know. So maybe it is like that. And indeed, some people, Menon group in India, they simulated it, and it seems that maybe it works. So, this is the end of my story, and let me first tell you that I very much indebted to my co-authors, Jean-François Joigny and Kate Sanford. We discussed it with a number of people here, and maybe I should tell you what are open questions. I told you how to deal with 2 particles. To deal with 3 particles turns out to be much harder, and we are still working on that. So this seems to be very obvious continuation, but we are still working on that. In equilibrium statistical mechanics, you know that everything is free energy. If you know free energy, you are the king of the event. No problem for you. In this out of equilibrium statistical mechanics, there are some analogies, but not complete analogies, and how far the analogy can go is not clear. Now, I told you that there is surface tension. I told you that there is phase segregation, but I never told you what is the interface. Phase segregation between oil and water is very flat because there is surface tension. How about phase segregation between active stuff and passive stuff? Would that be a smooth surface, or would it be fluffy thing, or will it be fluctuating, or what it will be? We don't know. The simulation which I showed to you at the beginning showed that it's, well, relatively smooth, but we don't know how smooth it is. Normal, if you just drop oil into water, then it does not immediately separate. First, it forms some sort of nuclei or droplets of oil in water. So there is some kinetics involved. What is the kinetics in this case of activity-driven separation, especially when polymers are involved? And what are hydrodynamic properties? When these active particles are moving, they generate some sort of flow of surrounding liquid. How that plays a role, we don't know. So therefore, it means there are too many things for us to know, and therefore we have to rely on students who should get involved in that and answer some of these questions. Thank you.
Smooth, like the fish that they are moving.
Yeah.
But then if it's active and they are constantly moving, what makes this interface?
Well, first of all, for fishes and birds and similar macroscopic organisms, there is the thing which experts in the field call quorum sensing. For some reason, many of these organisms behave like they prefer, roughly speaking, you can imagine that you sense, let's say, direction and speed of motion of 10 of your neighbors of different sides. You measure the speed, you average it, and you decide to move with the average speed of your neighbors. Right? This is called quorum sensing. Quorum sensing sounds to me like not physical property, but some property of these organisms. But then you can analyze consequences if your particles have this property of quorum sensing. Then, if you assume that, you can imagine that for particles on the surface of some cluster, they have fewer neighbors to average over, and you can make different assumptions about what they do. For instance, they can give bigger statistical weight to those neighbors whom they see, and then they align with those neighbors, or they may do something else. Depending on that, you can obtain any type of surface behavior you want, pretty much. So I don't know what to say about it and how real fishes work. In our primitive models with different temperatures, it should be easier, but we so far didn't do it.
Because in the experiment of Paul Chaikin that you showed, it seems that it was not smooth.
In the experiment of Paul Chaikin and Jeremy Palachi, it is even more tricky because what they see is more crystal phase than just a phase. So these particles seem to be forming regular crystal structures. Then outer contour is related to symmetric axis of this crystal. So it is a very peculiar story. It's not only surface tension, it's something, or you can say it's anisotropic surface tension. And as far as I know, this is also not well understood. Thank you.